Okay, good morning, everyone. I'm Marina Caskey. I am an investigator here at the Rockefeller University in New York, and I'm actually originally from Brazil, so Carnival means a lot to me, even though I am today in this cold New York City, not celebrating with everyone else. So what I'm going to be talking about today is about broadly neutralizing antibodies, broadly speaking, but really trying to work through why we are thinking about bNAbs as one of the potential cure strategies. So to start, I will just take us a few steps back to talk about these molecules, antibodies, broadly speaking. So the immune system, and in particular antibodies, they have these three main properties where antibodies are highly specific, and then highlighted here is a viral particle with these orange knobs are what a given antibody will be able to see and attach to. And the antibodies are these Y-shaped molecules. And in purple, this is the part of the antibody that is specific, meaning that it can bind to a specific, well-defined area of a given virus, for example. And at the same time, the repertoire of antibodies that our bodies are able to produce is very diverse. And that is what allows us to fight off so many infections that we are exposed to over our lifetimes and adjust as these viruses like we are seeing with SARS-CoV-2 evolve over time. So it's important to think about antibodies as being specific, but also the repertoire being very diverse. So the other part of the antibodies here in the bottom is what we call the Fc domain. And this is the part of the antibody that then can signal to the rest of the immune system to bring in other cells of the immune system to fight that given threat or that given infection. And then because of these different properties, antibodies can have different functions. So when we think about antibodies in comparisons with antiretrovirus, you'll see why they differ. So antibodies can attach to free circulating viruses and clear those viruses, preventing new rounds of infection. But antibodies, through their Fc domains, can also recognize, as illustrated here in this cartoon, infected cells that will express pieces of HIV, for example, on their surface, where that specific part can attach to the envelope that is on the surface of the cell. And then the Fc can signal to the rest of the immune system to say that that cell carries the virus and needs to be eliminated, needs to be killed. In addition, the antibodies can form these complexes with circulating viruses. And again, that serves as a signal to other parts of the immune system to come and clear that infection. And these are these properties that antibodies have that make them different from ART. However, in HIV, it's very challenging to develop antibodies that have enough diversity to cover the various strains of HIV that can exist. So in here, it illustrates three of these challenges. One is that these neutralizing antibodies recognize the envelope protein, which is on the surface of the virus. And that is the only target that is susceptible to neutralization, to being cleared by an antibody. And these proteins are sparsely, they are rare on the surface of the virus. So there's not a lot for the immune system to see to begin with. And in addition to that, what this spider chart here is trying to illustrate is how different the different strains of HIV are from one another. So you know how we receive a different influenza vaccine every year, and then the diversity of influenza viruses is illustrated by the small black spider web, in contrast to the different strains of HIV that were circulating in a given country in Africa many years ago. So this just illustrates how diverse HIV really is in comparison to even a virus like influenza, for which we need a new vaccine each year. So that illustrates how challenging it is for our bodies to make antibodies that really could recognize the diversity of HIV. Despite these challenges, there are individuals that are infected with HIV that during the course of their infection, they go on to produce antibodies that recognize different areas of this envelope protein. And this is what this picture here is illustrating. This is the envelope, the surface protein on the surface of the virus that can be seen by antibodies. And the colored areas are the different areas of this envelope protein that can be recognized by by antibodies that are broad, meaning that they have, at least in vitro, activity against different strains of HIV. However, not a single one of them achieves 100% neutralization. So that's why when we'll go through the studies, you see that we need to combine different antibodies so that we can increase the breadth of this approach. So I'm going to use the two antibodies that we have been working here at Rockefeller just to illustrate some of the points that we are trying to talk about antibodies in the context of cure research. So this is again a structure where the bottom part illustrates that envelope, what the human body, the immune system can see. And the red and the blue are the two antibodies, how they bind in different angles to this envelope protein. And then by doing so, they provide greater coverage, which is illustrated here in this graph, of how much of how a percent of viruses that were tested that could be blocked from causing infection when the combination in red of the two antibodies were given. What we would like to see is to have a combination of antibodies that would provide 100% coverage at very low concentration of the antibodies. And we don't have that yet, but with improvements, we are approaching coming over to this left corner. Together, these two antibodies, so even though they're not perfect, together, at least in vitro, they've shown to have activity against 80 to 90% of HIV strains that are meant to represent circulating strains in different areas of the world. And what have we learned from these antibodies when we've tested them clinically? So we've seen that when we gave either one or these two antibodies together in people with HIV that were not taking HIV medications, we saw that their viral loads went down for a period of time. In other set of studies, when we gave the antibodies to individuals that had been on suppressive ART, so they had undetectable viral loads while taking their medications, and then we switched them from ART to antibodies, the antibodies were able to maintain virus suppression for a period of time, at least in a subset of individuals. These antibodies now have also been modified so that they can last in the body after they are given for a longer period of time. So that will allow the studies to become less complex, where we could potentially just give the antibodies twice a year, for example. However, across these first studies, what we learned is that even though the antibodies showed to be very broad in activity in vitro, there was always in every study that has been done, a subset of participants that had resistance to the antibodies at baseline. And then the reason that we can that happens is you saw how I showed how the virus can be very diverse when we think about circulating viruses. But even within a person that lives with HIV, there is great diversity. So what this chart here is showing is this is a way to illustrate the different viral sequences, the different HIV sequences within blood cells of people with HIV. And then I chose these three because I think they illustrate how different one person can be from the other. So each of each of these trees illustrates one person with HIV. So if we look at this one first, 9252, you see this is a very narrow tree where in the cells, in the blood cells, in the CD4 cells of this person, there is only really one big family of HIV that could be re-stimulated to grow outside of the cell. So that was the reservoir of this one person. It was very, what we'd say, flat, because it was this very large clone of the same type of virus. And this is in contrast with this person here in the middle, for example, where you see that there are different branches of the tree. And the further away you are from the center of the tree, it means that each of these families are further apart from one another. They are different from one another. And the other part to this is that these numbers here that you see, for example, for 3BNC 3.7 and 1074.4, that says how sensitive this particular family of viruses would be to 3BNC or 1074. So this means that this is very sensitive to 1074. A little amount of 1074 would block infection from this family of viruses. Whereas you would need a higher concentration of 3BNC. But if you move down to the tree, you see that this family of viruses right here are more resistant to 3B and C. This one is less resistant, and so on and so forth. So I hope that this helps to illustrate how we talk about HIV being very diverse as it circulates in the community, but it's also very diverse within a person.
So Marina, may I ask you a question here? So
Yes.
do we know what causes this resistance? In other words, what makes certain viruses resistant to the bNAbs versus others?
So the reason this happens is from the moment that someone is infected with HIV, the body starts to fight off the virus, and then it starts fighting it off by producing antibodies. So the antibodies then push that virus to try to suppress the virus, and then the virus mutates itself. So those first antibodies don't work anymore, and then there's a new round of antibodies that causes a new round of viruses, and so on and so forth. So over time, until the person is put on ART, there is this viral evolution that allows from a few viruses that cause the infection to begin with that diversify and then lead to this reservoir of many different strains. And then the same way that the viruses diversified, the antibody responses against this diversity of viruses also evolved over time.
So this is a little different than what we call drug resistance, when someone has been exposed to a medicine and their virus then overcomes that because they may have been having challenges with adherence. It's different. It's a different situation.
It's a different situation. It's a different situation because it's happening naturally as we fight off any infection.
Right.
With ART, the person is only exposed to ART if you take on and off ART.
Right.
Here, the part that is challenging with antibodies to begin with is because even if a person does not have a broadly neutralizing antibody, that person has other types of antibodies that have been coexisting with viruses.
Right. I see. So we have. Okay. Sorry, I didn't mean to interrupt you.
Okay. So, all right. So then, so that is a problem because we know that the antibodies can work well, but we also need to identify for whom a given combination of antibodies would work well. And then the way to do that is to try to get a glimpse of what's in the reservoir, what's hidden in the cells or in the blood of these individuals. So there is an assay that was being developed, and what this tries to do is either from serum, so free viruses that are floating in the blood, or viruses that are within CD4 cells. In this test in the laboratory, they express the envelope, so try to extract what that given HIV looks like in terms of their sequence. And they recreate what seems to represent the majority of the sequences in a person, either in free-flowing the blood or within a number of CD4 T cells. And then they create a pseudovirus, so they insert that sequence of HIV into a tool that looks like a virus and then can infect a cell. And then they want to test if a given virus is sensitive or not sensitive to a given antibody. So if for a virus that is sensitive, if you mix it together with the antibodies would block this virus from infecting the cell, and the cell is not going to give a color. But if the antibody cannot block infection, then the virus gets into the cell, and that triggers the cell to show a color. And then we can measure how resistant to the antibody that given cell, to the antibody that given virus was. So this assay was tested in a study that Katie Barr led in the ACTG, where they looked at samples from plasma, so that's free-flowing viruses, or samples or CD4 T cells, so viruses that were in the reservoir hiding in the cells. And what's interesting and important about the study is that they saw that a given antibody would have similar level of activity against viruses that were found in plasma free before the person started ART, and 1 in 3 years after the person had been on ART within their cells. So it gave an impetus to develop this assay as a potential tool to screen participants for sensitivity or resistance to a given antibody. And so recently we applied this assay in a small study that we did with these 2 antibodies that we are working with. And what these plots here are showing, so each plot represents one participant in the study, where black is the viral load over time, and then the colors are the two antibodies that we gave. So as you can see in these 4 graphs, we gave the antibody here times 0, and then you see that there is a decline in viral load for a short period of time, and then that returns up to baseline, despite having plenty of the 2 antibodies on board. So what it means is that the antibodies had activity because it brought the viral load down, but that was short-lived. And when we looked at the results from these assays, we could explain that the reason that this was short-lived is because in this case here, the antibodies, the viruses were not sensitive at all to 1074 and had sensitivity to 3B and C, but like it happens with ART, if you have only one antibody, it gives room for the viruses to mutate themselves or to select for viruses that are resistant to this one antibody, and then the effect becomes short-lived. And then in contrast to that, if we had 2 other participants that, unlike these first ones, they had declines in their viral loads, and then they continued to have suppressed viral load for a very long time, as long as the 2 antibodies had been on board. And what's different between these 2 participants from the first 4 is that the baseline viruses in their plasma, so the free-flowing viruses, were very sensitive to the 2 antibodies. So in this context, this assay worked really well to us, even though it's a very small pilot study, and it would have helped us predict who would have a better response to this particular combination of antibodies. So so that's one type of study that we've done. Another type of study that I mentioned is that we have enrolled individuals that had been doing very well on ART with undetectable viral loads. And then we asked the question, can we switch them from ART and maintain virus suppression now that the viruses are at a lower level with repeated doses of the antibodies? So in this case, we gave 7 infusions of this combination of antibodies over a period of 5 months. And then we enrolled participants that discontinued ART at the beginning of the study or continued on ART while they received the antibody infusion. So the first part of the study is about, could the antibodies maintain suppression? And here I'm illustrating this a little differently. So in this, these gray bars here represent the individuals that started the study on suppressive ART, switched once they started the antibody infusions. And then you see that for some participants, within a few weeks, the virus returned, so meaning that the antibodies were not sufficient to maintain the viral load undetectable. Whereas for other participants, they were able to maintain the virus suppressed for an extended period of time. The last dose of antibodies was about at week 20. So as the antibody waned after the last dose, participants started to have the virus return to plasma. But in 2 of them, the viruses continued to be suppressed for an extended period of time until the end of study follow-up. So we could see that there are 2 different, or I guess 3 different ways to respond. Individuals that didn't respond and the antibodies were not sufficient, and individuals that maintained suppression as long as the antibody levels were likely high enough in the blood to keep the virus down. And then these 2 that were outliers and continued to maintain suppression beyond the study. So this study began before the PhenoSense assay was available. So we went back and looked at that assay that I described to you to ask, can we explain the clinical responses that we saw based on the results from that test? So, and this is illustrated here. So in gray, those are samples that we were not able to obtain a result from the PhenoSense assay, and we don't understand exactly why that is. So that's one drawback. And we saw that in some situations, for example, for this person 5105, he appeared to be resistant to the 2 antibodies, and then this correlated with the outcome. And then we had other situations where the person seemed to be sensitive to one antibody, not the other, but the person maintained suppression for over 6 months. And then we have these 2 individuals that maintained suppression for this very long period of time, but they, according to the assay, they were resistant to the 2 antibodies. So in this particular study, The use of this assay, which is a phenotypic assay because it measures the ability to block infection in vitro, this assay in this particular cohort, you looking at viruses within CD4 T cells would not have guided our patient selection or the outcomes that we saw. In contrast to that, Louise will talk about this later. They conducted a study at UPenn where they used the assay to bring participants into the study. And then they found that this same combination of two antibodies had very good response rate in individuals maintaining virus suppression for a longer period of time. So there may be some value for screening with this assay, but we just have to keep in mind that it's not going to be as black and white as we would like it to be. And the reason for that, it may be that because only few cells are being tested, that I'm just going to go back here, that when you have someone as complex as this, if the virus that ends up being replicated or being used to test happens to be this way, this one here, for example, it will look like the person is very resistant to 3DNC. Whereas if another virus would be the one that gets to be amplified and tested, it would look like that person would be very sensitive. So that is some of the caveats about this particular assay. But a lot of studies are applying the assay on clinical trials. And so we are going to learn what it can and cannot, how it can or cannot help us. So just to summarize this part, so antibodies alone or in combination can lead to declining viral loads for a period of time. When we have individuals that have sensitive viruses to both antibodies and we continue to give the antibodies over a period of time, we are able to maintain viral suppression in individuals that had been on ART and switch over to antibodies. However, we continue to have this challenge that there is this great reservoir diversity with strains in the reservoir that they may be resistant to the antibodies, and we don't have an ideal tool to characterize this yet. So switching over to cure. So can antibodies help cure strategies? So the barrier to cure is the HIV reservoir, which are the cells that I've been alluding to that have viruses within them that can reactivate once we withdraw ART. And we can divide this, well, there's maybe a third approach now, but there are these two main approaches where we like to kill these infected cells that harbor the reservoir, or we want to enhance immunity against these cells so that we may not eliminate every single cell that harbors HIV, but we are able to avoid further replication and then an eventual viral load rebound. So as I mentioned to you earlier, the antibodies can kill infected cells if they are able to detect cells that have viruses in them and from time to time may express pieces of HIV on their surface. And these antibodies can also bind to viruses that may be produced from time to time and then serve as an alert to other arms of the immune system so that these T cells can then come and suppress and kill cells that are still infected. We have seen many studies in non-human primates using antibodies alone or in combination with either vaccines or other immune modulating agents, leading to control of viral replication, even after the intervention was cleared. And in humans, there are still several open questions. For example, we don't know exactly what's the best timing for cure interventions. There is an idea that it would be best to intervene earlier because, as I explained it earlier, it's likely that that reservoir is less complex and there is also less damage perhaps to the person's own immune system and ability to fight HIV. However, it's hard to identify individuals early during HIV infection. Will interventions be more effective when they are added to ART or when they are given in the absence of ART? Will ART-free control of HIV require, in addition to bNAbs, as we saw in the non-human primates studies, combinations with other drugs that will, together with the bNAbs, lead to this better immunologic control or elimination of the reservoir? So what do we know?
May I interrupt, Marina? I just wanted to talk about that because I know one of the studies that was at Croy did show that the combination of using a latency reversing agent called romidepsin, as well as the bNAb, I forget what, BNC 3117, something like that, it actually created some post-treatment control, at least in one individual. And I think it was 6 or 7 were able to at least do an ATI for 3 months, I think it was. So that's what you're talking about in that last point.
Yes, and then I actually have a slide showing the results from that particular study just to illustrate. And yes, so that is alluding to that,
Okay.
where in addition to bNAbs, should we be combining them with other drugs or vaccines, for example?
Great.
So so I've been saying that, so far what I've showed is that the antibodies can bring the viral load down, and studies in animals and in cancer therapies also show that antibodies can improve the way that the body handles either cancer cells or viruses. So are we able to see this when we give HIV bNAbs to people living with HIV? Can we change these immunologic responses? So here it's looking at the T cell arm of the immune system. What we are looking at, this graph here is in a group of people that had not received bNAbs. And then we looked at the amount of cells against HIV at baseline and 3 months later. And you see that there's just no real pattern or change over time in this group of individuals. And this is in contrast to the participants that received the combination of antibodies after we withdrew ART, where we see, at least temporarily, once ART is withdrawn, the viral load continues to be suppressed, but mediated by the bNAbs only. You see that there is an increase of CD8 responses over time. And then as that effect goes down, these immune responses go back to baseline. So this type of change wasn't sufficient to prevent the virus from eventually coming back and rebounding, but it shows how during bNAb, something different was happening immunologically in comparison to when someone was on ART and there was no additional intervention. How about in the reservoir? Can we see any changes in the reservoir? So this is a study that we finished. So that is that study that I showed in terms of viral rebound, but now I'm showing the same study in terms of reservoir. So we had tried to measure at the reservoir, to measure the amount of viruses that are likely to be able to replicate if we take away ART within CD4 T cells. And we want to see if the number of those cells carrying those viruses would change over time in this month period before and after the person received the antibodies, either with or without ART. And in this assay, you're able to distinguish viruses that are intact, so meaning that the sequence is perfect, and so that virus has the ability to, if ART comes out, for that cell to produce a live virus that can then cause a viral rebound that you can measure in the blood, versus defective sequences. So these are pieces of HIV that would never be able to cause a new rounds of HIV replication because they are broken, they are not intact, they are called defective. And that is actually the bulk of the HIV that is still hidden within CD4 cells. But it's this minority of intact viruses that are the reason why we can cure or control HIV without treatment. So when we make this distinction between intact or defective, and we focus here on the defective, we found that in this group of, and this is just a group of 13 individuals that had been on ART for at least 7 years when they went on this period of bNAb therapy, we see that there was a decline in the number of cells harboring these intact proviruses. But you see that the difference is small, but it was there and it was significant, and it was different from what we saw in terms of defective viruses. So the antibodies didn't seem to interfere with the broken down, the irrelevant defective viruses. And in a parallel cohort that had not received the antibodies, we also don't see a change in either the intact compartment of viruses or the defectives. So this, again, it's like the immunologic data that I showed, is an indication that perhaps when we give the combination of antibodies for a longer period of time, there's something happening in the reservoir that we are not able to see with ART alone. But it's a small enough effect that didn't prevent rebound from happening when ART was withdrawn. So how about combination? So this is a cartoon from an IAS paper that Steve Deeks led that illustrates the various types of interventions that can be used in combination during this period of time when someone is with suppressed viral loads, with the idea that if ART is to be discontinued, there may be a period of viral rebound, but the goal would be that this would be followed by virus suppression and the person not needing to go back on treatment. And it's this, what is the ideal combination and the ideal subset of participants for such studies that we are trying to investigate, we meaning the entire field. And this is a study that you alluded to, where an antibody was given with romidepsin. So romidepsin is a drug that can stimulate virus out of hiding, so outside of the cells. Even when someone is on ART, it can cause the cells to produce a little bit of virus. So the idea here is that romidepsin would push the virus out of these cells so that they would be more visible to the antibody that would then clear. And then in this study, what's interesting is that the interventions were given at the initiation of ART. So the goal was to see if we give an antibody together with this molecule that pushes the cells, the viruses out of the cells, as the person is initiating ART, can we interfere with the way the reservoir, the viruses settle down in the reservoir, and in how the immune responses against that reservoir are over time? And will these modifications perhaps have affect how this person, these individuals behave after ART is discontinued? And then these are the different groups of the study. So they have three major observations. One is that the rate of viral load decline over time was faster. There's this very fast first phase of viral load decay, and then it kind of slows down. But the two groups that received the antibodies, they had faster decay. So they achieved undetectable levels at a faster rate than the groups that did not receive the antibody. And then this was even more clear in the subset of participants that were in fact sensitive to this particular antibody. So that's one observation. The other one is that they looked at CD8 T cells at start. So this is a group that received just ART, and then these are the groups that received the antibody with or without romidepsin. And at the beginning, there is more CD8 T cells because there is virus. The body is fighting that virus to bring it down. And once the viral loads are undetectable, the amount of these T cells decline because there's less virus. ART has suppressed viral replication. And this is what happened in the ART only group. In the group that received the antibodies, you see that early on there is a lot of CD8 T cells, and then it declines a little bit by 3 months. But at 1 year, it's still higher than if we are to compare to the ART only group. And this is more evident or only evident really, in the subset of participants that had sensitivity to the antibodies. So again, this suggests that there's something immunologic that happened early on as the person was viremic, starting ART, received this intervention. Somehow it seems that it set the immune responses against HIV in such a way that they were preserved and high over time, even on suppressive ART. And then finally, when a year later they discontinued ART, these are the participants that had received the antibody. And then this is illustrating time until viral rebound and then the level of viral loads. So the blue participants are the ones that received the antibody and had viruses to begin with that were sensitive to the viruses. You see that there is a delay until the virus returns. The virus doesn't seem to peak as high before ART is initiated. And this is the one participant that you alluded to that continued to maintain suppression for a long period of time. Even in the absence of any treatment. So again, this shows that this intervention at this early stage of ART initiation did something immunologically and perhaps to the reservoir that was different than ART alone. So this is just to summarize that in terms of cure strategies, we have initial data showing that HIV-specific T cell responses can be enhanced during bNAb therapy, either during ART interruption or at ART initiation, and also the antibodies may interfere with the size of the reservoir. So just to wrap it all up, I didn't talk about this, but the AMP studies showed us that antibodies can in fact mediate protection from HIV as long as the antibodies used have activity against the infecting viruses. Antibodies can maintain virus suppression when ART is discontinued and the person switched to the antibodies. We have very early evidence that perhaps antibodies can impact the intact proviral reservoir and modify immune responses. However, we continue to struggle with the fact that there is pre-existing resistance within people that are particularly chronically infected with HIV. And then we have to really develop good tools to identify what antibody combination might work for a given individual. And I don't have time to go into this, but there is a series of studies that are planned that will look at different combinations, different clinical settings, and even different ways of delivering antibodies to continue to add to our knowledge in terms of how antibodies can be useful in the context of HIV cure research.
Mm-hmm. Well, thank you. Go ahead. Go ahead.
Outstanding presentation.
Yes, great. But we had some questions.
Yes.
One that was really sort of coming up time and again was, do we think that these bNAbs are going to be available to everybody, or do you think it's just going to be a select chosen few?
Is the question about cost or
access and cost? Access.
So that is a big problem for sure. And that's why there is an interest in, the antibodies have already been modified to be long acting. So then the amount required becomes much smaller. So then that decreases cost. And there's also groups that are looking at different strategies to deliver the antibodies in a way that it can be self-renewing in the body, either to viral vectors that would cause cells within the person's own body to produce that antibody. And if these strategies would be successful, then those antibodies it would be like vaccination in a way, where those antibodies would be able to be produced over a very long period of time. So right now they are way more costly than ARVs, but there is an understanding that really, if we are talking about HIV cure research, we need to be thinking about low-income countries because that's where the burden of the infection is. So there is, while we try to understand what bNAbs can and cannot do, there is another set of efforts to decrease the cost and to think about these clever ways of delivering antibodies there for a longer period of time so that the cost can decrease.
Awesome. Thank you so much for that. Yes. We have a hand from Rowena Johnston, and then we'll hit back the chat to make sure we've addressed all the questions.
Go ahead, Rowena.
Thank you. Thanks for that, Marina. I know, there was a lot of material in there. I wonder if you could talk a little bit about the contrasting results between the Roadmap trial and the eClear trial, because they used the same interventions and had very, very different outcomes.
Right, yes. And, I had the Roadmap study, but there was already so much that I took it out. So Roadmap was a study that we did with Rowena in MFAR, where we gave the same combination of 3-BNC and romidepsin. But this time around, we gave the intervention in people that had already been on ART. They were chronically infected, and the intervention was given during ART suppression entirely. And like eClear, we discontinued ART after the antibodies had cleared. So in that study, we didn't see any changes in terms of reservoir size or immune responses or really time to viral rebound. So it's very striking, the difference between the two groups. And we think that this really has to do with the fact that it speaks to the fact that the antibodies had this opportunity, so to speak, to interact with the virus and then perhaps form immune complexes and do what we call a vaccino effect. So really improve immune responses as they were forming at a time when ART was initiated. So ART was bringing the virus down, but there was still enough virus to allow for this antibody viral interaction that we think helped establish perhaps a less, we don't know that yet, but a less diverse, but for sure better immune responses that might have mediated some of this immune control that we see later on.
Awesome stuff. Michael, did you want to hit the chat?
Yes, so there was a lot going on in there. People were asking about testing. and I'm trying to flip back into that. I think we've answered the question about PrEP, using this as prevention. So people were wanting to know, do you test against all the different isolates from the different regions? And do you think we could pre-screen participants for bNAb sensitivity? I think we've already kind of answered that a little bit, but they were talking about two different approaches that were brought up at CROI. In
terms of how to test for the antibodies?
Yes.
Yeah, so there are different, you know, there is, I think it was the VRC, they had a method that it was by outgrowing viruses from cells. And then this other one by monogram, which is trying to find the sequence and then creating the pseudoviruses. And I think they both are helpful to some extent, but you have to remember that when you're doing either assay and you have 5 million CD4 cells, right? It's a small number of CD4 cells, and within those, you may have a handful that carry the virus. Mm-hmm. So it may be that depending on how diverse a person, the diversity of viruses that a given person has, that you're not going to, by chance, end up with the representative viruses within that small number of cells. And I think that is probably the truth about either assay. And then, but they're improving in the way that they are done. So that's one issue. And then the other issue is for some antibodies, for 1074, for example, so it's very clear what the mutation is. So it's very black and white. If you have these mutations, like for ARV, that antibody is not going to work for you. For other antibodies like 3BNC117, it's less clear exactly what combination of mutations would make a virus sensitive or not. So for those antibodies, it is harder to really understand, even when you get the sequences, to really define resistance or sensitivity. But like I said, these assays now are being launched across many different clinical trials. So as we get clinical data back, we're going to be able to regroup and look at the results from the tests with the clinical outcomes, and then see how we can improve upon that.
Thank you for that. And thank you for this great presentation and telling us a little bit more about bNAbs.